우리들 만나고 헤어지는 모든 일들이 어쩌면 어린애들 놀이 같아. 슬픈 동화 속에 구름 타고 멀리 나르는 작은 요정들의 슬픈 이야기처럼, 그러나 우리들 알지도 못하고 울지만 사랑은 아름다운 꿈결처럼 고운 그대 손을 잡고 밤하늘을 날아서 궁전으로 갈 수도.
We are going to try to understand how to detect the specific fragments of interest. Let's take the example of this gel, where you have so many different DNA fragments here. So these are all the DNA fragments present in smear. And let's say you may have some specific target DNA bands that you want to identify, but it is impossible with this so many different fragments to pinpoint the presence or absence of your specific DNA or RNA samples like that. Then here comes the concept of a probe. Using the probe, you can specifically identify whether you have it or not, because once you let this whole sample interact with, you apply some specific probe, and then this probe can only detect by interacting with the specific target DNA fragment, and all other noise will not show, because they are not going to interact with the probe. So this interaction with the probe is the key. Probe will allow for specific interaction with only specific target of DNA based upon the sequence complementarity. So what you can say about the probe is, okay, what's probe? Probe is a small DNA fragment. Sometimes RNA, you can use RNA, small RNA fragment, or also can be used as a probe, but since it is practically more difficult to handle, so generally DNA has been the choice as a probe. And if you want to use any specific probe which can detect the presence of your target, it has to be complementary in the target sequence. So probe is a small DNA fragment having complementary sequence to specific target DNA or RNA. So this target, whether it is a DNA or RNA, it has to be made into single-stranded, so that this single-stranded area can interact the single-stranded DNA probe having this complementary base pair. So in other words, let's say, okay, you have this target DNA, double-stranded usually described as a DS, double-stranded DNA target. And to generate a probe which is specific to this double-stranded DNA target, it doesn't have to be, your probe DNA doesn't have to be the same length as the probe. Probe also does not have to be in the same length. No need for same length as the target, right? Probe can be even smaller than. So that probe, if you want to generate, so you have original target. Then you make your target single stranded. The way you do is by, for example, heat them. If you apply heat, then your target DNA will become single stranded like that. Then you apply your probe. And probe doesn't have to be in same length. So let's say your probe DNA small, and then this probe can interact the region of some part of the target DNA. So then they can have, instead of the original complementary base pair interaction, now taking this opportunity, then the probe DNA can sneak into to replace this complementary base pair into the relationship. Although this probe doesn't have to be in the same length as the target DNA. You have this complementary base pair in region over here. Now the question is how to visualize this interaction. You can use a radioactive isotope in preparing your probe DNA fragment. You can use the radioactive nucleotide. Then your probe DNA fragment will be a hot radioactive DNA fragment that can be easily detected by some instrument. Or in these days, actually, trying to label if this is your probe DNA, then you have specifically label your probe DNA with the fluorescence chemical. Like, it's called the fluorescence labeling. Either way, so that either you use the fluorescence or the radioactive isotope. Now the point is, now you can know where is your probe DNA. And in this complementary base pairing principle, where the probe present, then you can assume that there is your target DNA also that held together through this complementary base pairing is the key. So among all this different mixture of the DNA sample, if probe stick with the sample, then actually there is your target DNA. If you treat the sample with the probe and then wash them, and then if it doesn't show the presence of the probe DNA, that means there is no target DNA. It's that simple. So that's how you use your probe. So this little figure describes the basic principle of how to use the probe DNA. So this is a probe as a short fragment. Once again, the point is, you don't have to use the probe which is the same length as your target DNA. It can be much smaller. The only thing matters is whether they have this complementary base pairing relationship or not. So among these different DNA fragment in the original mixture of the DNA sample, let's say fragment A, B, C, and then the way you can assure that there is actually your target DNA, even though these DNA sequences may have a little bit of a very similar in DNA sequence, but specific the region that can form this complementary base pair only present in the sample C, so that probe will interact only with the sample C, and these two other irrelevant DNA fragment will not show any presence. So the point is, as long as this sample C is there, then it will show a positive signal. If the sample C is not there, then you won't see any positive signal. That's how you can test the presence. This little different techniques I will skip, although it is a big part of actual practically how to once you complete the gel electrophoresis, how to actually generate your DNA sample, convert them into single-stranded so that they can be ready for this hybrid. It's called hybridization. Which means interacting your target DNA samples with the probe, and so both of them are in the state of single-stranded. So that they can have this opportunity for the complementary base pairing, and then finally you can detect whether they have it or not, like this technique. But I won't go into, and I will not try to describe in lengthy ways. So basically all is not really necessary for you to understand the entire process, the logic. So the only thing important is that as long as you can understand this concept of a probe, and that's it. So I will simply zip through and go into the next session. So using this, you have everything now that can make possible for these techniques doable. So now here comes the DNA fingerprinting. Actually, originally it was invented by the scientist Alex Jeffrey in Great Britain. So he can be recognized as the pioneer of the DNA profiling. Is still alive and active in scientific research. So he discovered some repeats area, tandem repeats. Once again, this tandem repeats, and which this hyper variable means the sequence, like how many repeats are present in terms of this number of repeats, are highly variable among different individuals. And that was actually in the area around near the human alpha globin genes in early 1984. Here, this is also not necessary, so don't worry about. So the point is, where probably is much better. Let's say this is the whole DNA, okay, and if this was the area occupied by the alpha globin, okay, and to give you in a much simpler terms, he discovered around from this alpha globin gene area, he discovered some area which is DNA sequence shows in such a tandem repeat. So the DNA sequences appeared in many multiplicate of short sequences in repeat pattern, like for example AAGGCAT, for example, and this repeat appears in 5 times in individual A. On the other, and the same GGCAT repeat appears in 7 times in individual B, something like that. So he called that such a variable repeat area as a VNTR, standing for B, variable number. So in different individual, this number of repeats are different. That's why it can be called a variable number of tandem repeats, VNTR. So in DNA fingerprinting, this repeat area, something called the VNTR regions, are one of the very popular choices for using this and trying to identify the different pattern of different individual in terms of their DNA sequence. So VNTR is one of such popular repeat elements present in all human DNA. So whenever you hear some story about DNA fingerprinting, and inevitably you will get to hear this term VNTR. So you may wonder, what's VNTR? The point is, it's simply just a number of repeats, some of the specific DNA sequence repeats present in all different human, and this is one of the very popular choices for having this DNA fingerprinting. That's it. So this is one very simple example of using this VNTR allele in 6 different people. So the fragments and each different fragments and lengths are all different, like this photograph shows. This is one such gel electrophoresis picture. And how do you see these different fragments in length? Is when you can find some specific restriction enzyme that can cut the region flanking this repeat, then that's how you can generate different sizes of such different repeats in VNTR, for example, like here. In one individual, you have 3 repeats like this. So these repeats are in box like this. And another individual, you have 5 repeats. The way you can distinguish 3 repeats versus 5 repeats is if you can find any enzyme. This is a restriction enzyme. That can cut this region so that preserving the length of the repeat containing region. And perhaps the same restriction enzyme site is also there in the individual B. So if you apply the same restriction enzyme like this, then the length of fragment encompassing this repeat will be much larger. Okay. So if you, after cutting your whole DNA sample with this particular restriction enzyme, then you do this gel electrophoresis. Obviously, you will have to apply the probe. Okay, so in this case, probe, probably probe you can use as something that, for example, a part of this DNA, like this, it's just enough so that this probe DNA can interact with this sequence also like that, and that will be just simply enough to make and then to reveal the difference. So in the sample taken by the individual A sample, the fragment size would be, which was revealed by the probe interaction, would be something like in the intermediate in size. So the gel will migrate toward this. This will be positive and this will be negative electric pole. But on the other hand, individual B, which this restriction enzyme cutting pattern would be larger. So if you can detect the size of the fragment with this probe, then it will be larger like this. So that's how you can see the difference in, let's say this is one of the such VNTR repeat region. That's how you use this VNTR region in distinguishing different individuals this pattern. Right. So in sentence, what you can say is using a probe recognized a specific sequence within the repeats, so you can distinguish the different repeat number by the size. And this is how, so like in person A, once again, in person A is one such imaginary repeat. Let's say this is one example of a VNTR. Then this homologous chromosome, here this is a heterozygous. In terms of this locus of this VNTR containing locus. So in one homologous chromosome you have 3 repeats, but the counterpart homologous chromosome, other homologous chromosome, this number exists 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10 repeat. It can happen because one came from a mother side and the other one came from father side. So there is no reason why this repeat, because different individuals will have a different repeat number originally speaking, and your mother and father were 2 different independent humans. So there's no reason why you expect that they would have exactly the same number of repeat in any given region of the entire chromosome locus loci. But on the other hand, person B happens to carry the homozygous status. So number of repeats 1, 2, 3, 4, 5, 6. In both homologous chromosome pair, this person B has 6 repeat. So the logic that I have just explained, if you apply this particular restriction enzyme which cut this flanking area, then person A will generate two different fragments in different sizes. One is larger and the other one is smaller. On the other hand, person B will generate only one intermediate size of a fragment like this. DNA fingerprint application. And especially in once again, if you can understand this type of a so-called VNTR, are spread all over the places in different location of chromosomes. So by combining all of this data, you can generate very, very unique paternity one individual case. Such, so in either way, whether a child is whether truly genetically related, the children of the particular couple, whether it's is this the child of this father, or is this a child of this mother? Like, we have this real example very recently in a certain part of this country. A baby, whether it belongs to a grandmother or a mother, DNA sequence, DNA testing, the accuracy of this DNA testing, nobody can argue against this. So the person still maintaining that the DNA testing is inaccurate, so you cannot give any 100% credibility to the test result. Is absolutely out of she does not know what she's saying, because lack of this information, she can argue such a very stupid claim. But so it's not about DNA sequence, actually DNA sequencing, but usually it's done by testing this how many repeats and that does that repeat pattern matches. So this is a very simplistic such a scenario of an example here, in that let's say this particular sequence GGA, GGT, GG, whatever this sequence called imaginary sequence, and this sequence probably would present in many different locations of the whole entire genome. Like here, three loci in locus 1 in one particular chromosome, and the other location locus 2 of another chromosome, and locus 3 of another chromosome. So there are three loci that have exactly the same this sequence in repeats is the question. So so in the case of this particular mother, and this one particular locus, in one homologous chromosome, there are two repeats of this sequence, and the same location, but on the other homologous chromosome, the number of repeats is 5. And another locus number 3, this particular DNA sequence repeat exists in 3 and 7 in these two homologous chromosome. And finally, on the third location of the chromosomes, the number 3 loci, the repeat number is 1 versus 8. This is how this repeat sequence is present in this particular model. And if there was a case, a biological child of this mother will definitely inherit either one of these on this chromosome, either this or that. Either one of these will end up in the child chromosome, because during this gamete production, either one of these two homologous chromosomes will be picked up and then go to the one particular gamete, and that gamete will later fertilize with the father's gamete, and that's actually what you are, the identity as a child. And the same thing, so either among this number 3, the repeat number 7 versus 3, either one of these will be present, and same thing. So if you compare this DNA fingerprint pattern with the mother, okay, and also father, and this child's pattern, well, exactly, we do not know the pattern of this father because it does not show over here, but at least half of these two, at least exactly the half of the mother's fingerprint pattern will be present, and another half would come from exactly from that of a father. However, if this child is not actually the one related with this mother and father's marriage, and so like an unrelated case, this pattern will not match either one of these half of the mother or father's pattern. And that's how you can distinguish. And you may think, okay, that is too simple, so how can you by using this logic, how can you distinguish all of the different individual cases as a like a unique pattern? Don't you worry, this was only the example of 3 locuses, but there are so many different locuses over many different unique this repeat pattern. So no single person, no two person, I mean what I mean by this, no two people will share exactly identical such fingerprint pattern revealed by this probe, some specific probe, is the whole idea. So this one, although you may not actually see some what's what, but these are so-called like a multi-locus DNA fingerprint from a large family using one probe of a GTG 5 times repeat probe, and you have these, even this highly related large family. Actually, if you look closely enough, then not a single such pattern is identical with any others. So that's something that you can be assured. So DNA fingerprint can be utilized in identification of a unique individual, is the point. So there are so many such different types of repeat present, and there is no point of going over exhaustively like each individual different types of repeats that used to be or can still be used in these days. This piece of information is just for this particular bioscience majors who are kind of still interested in the differences among different types of such repeat DNA and what's used in which case. So if you happen to be one of those students, then you might want to actually really go over and read so that you can have a piece of information. Other than that, you don't have to worry anything about this. So I will just simply zip through so many different types of DNA repeats, including something called the satellite DNA and what's what, and mini satellite, and also microsatellite and whatever. And actually the VNTR, that famous VNTR, is a part of this so-called microsatellite. And also not only VNTR, some other such important like such repeat elements named STR or SSRs are also part of this microsatellite or whatever, right? And yeah, so like that. So for example, using such this particular repeat called STR in human chromosome, there are 13 specific already in the FBI of the United States in their application of a forensic science in identifying different individual. They have already prepared 13 specific sets of such specific DNA sequence elements, all together combined with can be generated a unique pattern of an individual, like in the chromosome number 2, 3, like, and number 4 you have, and number 5 you have 2 different loci, something like that. And then finally, whether that sample belongs to a female or male, you have also gender specific the marker are also present. So this is such a one practical example of such a usage of particular repeat DNA sequence in DNA fingerprints. Yeah. Okay, with that, yeah, I think this DNA fingerprinting and this profiling, the description of them are complete. One more thing is, although I'm not a Tim Cook or Steve Jobs, there's one more thing is, so in practice in these days actually, you do not need to use restriction enzymes anymore. Like, it's not really convenient. You treat your whole DNA sample and then use some very specific DNA restriction enzyme to cut them and then run the gel. It's not really very convenient, relatively speaking, anymore, thanks to the newly available technique called, guess what? PCR. Yes, I mean PCR. There are so many different areas of application, and that's why the PCR is such a huge impact on the advancement of the pure molecular biology, as well as all other medicine and forensic science and so many things. Basically, what this PCR technology is, PCR, abbreviated from polymerase chain reaction. Polymerase is here. What this polymerase means is a DNA polymerase. Hey. So this is an enzyme catalyzing the replication of DNA. Like, let's say if this is the DNA double helix, and DNA polymerase will convert this into two copies of identical, so duplicated version of DNA. That's what the DNA polymerase does. Right? But here, using DNA polymerase, you are making this DNA duplication, DNA replication, chain reaction of DNA replication. Here, so here originally you have this single copy of a DNA fragment, and if you go through many rounds of DNA replication, then you end up something like 2 to the 10th, 2 to the power of 10th copy, which is tremendous. You cannot even count. Countless copies of DNA. For example, okay. So, but DNA replications for DNA polymerase to work, it requires a primer. What it means is, let's say you want to duplicate this, and DNA polymerase can only duplicate. It's really it's not really easy to say, but when DNA polymerase work, in order for it to start, you have to have first you have to separate this into two single-strand DNA to open up, and then there is some specific element called a primer. Primer is practically very similar to probe, right? Because between primer and the target DNA to be amplified, to be replicated, there should be a complementary base pair relationship should exist. And also, just like the case of a probe, a primer doesn't have to be entire same length as the target. Primer is actually is a very short DNA sequence fragment, and only in the presence of this primer, this DNA synthesis can now go. Maybe I should. Now DNA synthesis with the existing primer, DNA synthesis can go now like this in this 5 to 3 prime direction. So this directionality is so important. That's why I had to introduce this 5 prime end and 3 prime end of DNA. And since DNA is always arranged when in speaking of DNA double strand, their arrangement is always anti-parallel. So the original target DNA, the orientation is like this. This one is a 5 prime and this one is a 3 prime, and they are complementary. For the synthesis of the other strand, theoretically speaking, the primer should be placed the other end because their direction is different, and then synthesis should go to this direction. So this gotta be 5 prime end and this gotta be a 3 prime end. And original DNA sequence the 5 prime 3 prime end was in this orientation. Although in actual DNA synthesis, DNA replication in the cell, the things are different from what I have just described, but never mind. This is theoretically speaking. If I gave you the real story, it will be more confusing, so I will not touch on this. So basically the PCR technology is using these two primer sets. You are trying to amplify. So let's say this is your whole DNA on the chromosome, and if this area, if this area is your target, you want to amplify. This amplifies your mission using this PCR technology, because PCR technology is basically in this running this DNA replication in several rounds of in succession, like usually you about 30 times of repeated DNA replication reaction. Then as a consequence, this particular region is highly amplified in its, like how many? In each round of such DNA replication, you will generate double it. So if you repeat this DNA replications 30 times, then originally starting from one copy will end up to the 30s. How many of the 2 to the 30s copies of DNA replicate DNA sample? And that's the power of this PCR technology. You can amplify in huge number, and that's how you can identify some of the small traces of the sample on crime scene. Using this PCR technology, you can amplify in huge number, which are enough for the analysis. Same thing for the coronavirus detection testing. Small, very small, tiny bits of the RNA, as the coronavirus is basically is an RNA virus, and you can detect the presence of the RNA virus sequence using this PCR technique in huge amount, so that you can be certain clearly with confidence that whether this person has these traces of this RNA virus sample or not, is the whole idea. Anyway, the way let's say this is a target area now, this target DNA. You can, using this specific primer, running opposite way, these two primers, two sets of primers facing opposite direction, okay, and run this PCR technology. Then this area flanked by these two primers can be amplified in huge amount, is the whole principle of this PCR technology. So using this, instead of applying any specific restriction enzyme, in identifying, let's say you have this repeat, person A, you have 5 repeats. person B eight repeats. In the past, to test the difference between these two individuals, what you had to was using the restriction enzyme that can cut this area, right? The restriction enzyme. But now, instead of using this restriction enzyme, what you can instead use is primer, PCR primer, which PCR primer in yellow, okay, PCR primer having this area, and PCR primer having the complementary sequence with this area, and then the PCR reaction will generate this amplified region of DNA over here like this. This is a PCR product versus in this individual the PCR product will be basically like this. Actually, to be more accurate, the PCR product should be this, including the primer area. What are you doing? Like this, and this is the amplifier. So clearly you can see the difference in fragment length without using the much more inconvenient the restriction enzyme is the key point. So in these days, much more quickly using this PCR instead of restriction enzyme, the DNA fingerprinting can be done easily. That's the point. With that, so we finished the description of the technical aspect. The whole thing began with the first introduction of the concept of the heterochromatin. Now, so since we brought up this heterochromatin and euchromatin, now is the time to dig in a little further about the structure of what's the difference between euchromatin and the heterochromatin, like euchromatin versus heterochromatin, because their relative structure in the condensation is very important in the functionality in terms of the activity of the genes included in the region. So chromosomes and chromatin, already actually the difference between chromosome and chromatins are how high or how tightly are they condensed. So why chromosomes are the maximally condensed structure that can only be seen in the very brief period just right before the cell division, whereas the chromatins are relatively loosely organized structure in terms of its condensation. But both of them, whether it is a chromosome or chromatins, both of them are bound with this particular protein, the histone proteins. And the basic unit of this organization is called the nucleosome, right? So this is the in nucleosome, the histone proteins are the one actually that coordinate this relative degree of condensation. And I believe I have already shown this photograph. So this is the difference between the chromosome and these the chromatin. So difference is the same DNA, but this difference in how tightly you wrap the DNA around this, something is the difference. And this is the basic structure of such a nucleosome, in that 8 histone proteins of 4 different kinds form a core unit of this nucleosome, and DNAs are wrapped. And by adjusting the relative distance among nucleosome, you can achieve this degree of condensation. So the chromosome is the most condensed form, but in other period of the cell, actually, these chromosomes exist in as a chromatin, which is less condensed. But there are 2 different kinds of chromatin. One is heterochromatin, which is relatively speaking more condensed. Of course, it cannot be compared with that of the chromosome. Chromosome is the most highly condensed form, but this heterochromatin is also like a very tightly condensed in its structure. And especially in the case of this constitutive heterochromatin area, this region represents a very gene poor area. You seldom get to see the presence of genes, and as I already mentioned, they are, in terms of their regions, specific regions, the centromere and the telomeres are such the constitutive heterochromatin area. On the other hand, euchromatins are rich in the genes. Many genes are there, rich in those areas, less condensed. So probably you can summarize in graphics. Heterochromatin is more condensed, and also that I'm going to explain this. Silence genes methyl. What is this? I will get to that a little bit in a minute. And heterochromatin areas, if you apply some specific staining on the nucleus, they are the region actually pick up this staining better, so it stains darker, whereas this euchromatin areas are lighter, and many genes are there, and those genes are actively expressed. Here the point is, except for those already like set up fixed permanent, so-called permanent or constitutive heterochromatin area. Except for that, a given region on the chromosome can switch back and forth between hetero and euchromatin is very interesting point. So, like here, euchromatin area, like I said, the distance between the nucleosomes are like larger, and whereas this heterochromatin area, this distance like these nucleosomes in a same area of the chromosome, so are packed, more compact, and the gene's activity, gene expression activity is like here in euchromatin area, the gene expression what do you mean by the gene expression? It is like how actively the transcription is RNA making activity. So if we define the gene as an area that can make RNA as a broader definition of genes, then their activity is very like high. They are actively engaged in this RNA making process, whereas this heterochromatin area, those activities just shut off. So this is a silence area, and the point is, any same region of the chromosome can switch between euchromatin structure and heterochromatin structure. And this type of actually taking advantage of such a relative chromatin condensation and trying to control the gene expression, the concept is epigenetic control. So this gene expression is first definition because this concept of epigenetics is newly emerged concept. Even during the time of my graduate study, this was beginning to emerge. So even before that, no one knows anything like this, such thing like epigenetics exists or not. But then these days, everybody recognize the importance and the widespread nature of this epigenetics. So we better actually at least understand what they are. Epigenetics, by definition, although there are a little different version of the definition do also exist, but the standard definition of this epigenetics is changes in trait, once again this trait, without DNA sequence alteration in the gene. So this concept is in contrast with the classical genetics concept established by the Gregor Mendel. So according to him, all different traits are the one generating different alleles, right? So why do you have different alleles of one single character or single gene? Because of the DNA sequence variation. So according to the basic classical or standard or traditional genetics concept, all trait variation, all variants in traits should be the result of DNA sequence change. So if you don't have any DNA sequence change, then you don't have any trait. Although something called acquired the traits, sometimes these acquired traits are observed, which is the result of environmental adaptation, but then these are discounted as a not true trait, because these are not going to be inherited into the next generation, because it does not, any acquired trait does not touch the DNA sequence. So it is something that not involving the DNA sequence change. But then later, scientists discovered something very strange. There is obviously differences in traits, but then if you compare these two individuals having two different traits coming out of one single character, but then there is no DNA sequence variation between these two individuals. But then are they the result of this just an environmental changes, like acquired trait? But then this case, it turns out that a lot of these are also something that are inherited into the next generations. So what is going on? And this epigenetics are like, how can you have any trait, some different trait, without involving the DNA sequence change? It can be achieved by the histone nucleosome modification. This histone modification can affect gene expression. Like heterochromatin, there is the link between the heterochromatin region. There is a reason why there is a gene pool, because those area, it is very difficult for any enzymes for gene expression have any clear, easier access to the target and make the RNA product. So that's why in the heterochromatin region, genes are silenced. You don't have any. If ever, you have very, very, very little gene expression. But euchromatin area is highly active because the nucleosomes are loosely organized, so you have much easier access for those enzymes involved in this gene expression. So genes are actively expressed. And then how can the difference in the gene expression level make different trait? One very good example, such an obvious example of this, like difference in gene expression level can make different trait, is, let's say, for example, growth hormone. If you get to express high amount of growth hormone, and as a consequence, you will achieve a bigger body mass. Whereas if you somehow are not able to express growth hormone, they are all the result of this gene expression for the growth hormone gene, then as a consequence, you will have a smaller body mass, like with the shorter statue, including such things. So this is only coming from differences in the gene expression, but obviously the consequence is two well distinguishable alternative trait. And this gene expression, differences in the gene expression, can be achieved without altering any DNA sequence. It's not something that result of a mutation, but without having any mutation, you have a certain individual have a higher gene expression and certain individual on that particular gene has a very smaller, lower level of gene expression, and as a consequence, a trait difference can be made. Chemically speaking, I'm not really sure whether I even have to mention this or not, but simply it's just such a so well known and well mentioned, frequently mentioned phenomena in these days. So even laymen like us probably should know about a little about this. So this chemical modification is the key. Like some two different chemical modifications like acetylation versus methylation. These are the two examples of a type of such chemical modification. So if the histones on the particular area of the nucleosomes are like the acetyl group of these particular chemicals are attached to this histone, and then this chromatin area will become much less condensed, and then it will become euchromatin, and then as a consequence, genes over there will be activated. Like activated means higher amount of gene expression will be there. So this is a chemical structure of acetyl group, which you don't know. You do not have to know the structure, but the point is, some particular part of the histone protein, sometimes by the action of the enzyme, this acetyl group can be attached, and as a consequence, then these histones on the nucleosome can exercise a much loosely organized, like a more of this social distancing will be displaced, and as a consequence, then gene will be activated like this. If you have an acetyl group attached, then suddenly the average distance among different nucleosomes, like here, will have larger distance. On the other hand, maybe it's too general, but generally speaking, putting a methyl group is this, methyl group on the histones, then will reverse this status, and those areas will become heterochromatin, and as a consequence, gene activity, gene expression will be shut down. Yes. And this acetylation versus methylations are all by recruiting some very the effective proteins that enabling such chromatin modification. Yeah, so and this, what kind of factors can affect in a given time this particular area of the chromosome become heterochromatin or vice versa? In many cases, it turned out that some specific environmental conditions can influence this transition between switch between heterochromatin and euchromatin on any particular area of the chromosome. Okay, so this is something that a lot of those epigenetic traits appear to behave as if they are an acquired trait. So apparently you don't find any DNA sequence change, but then it is not actually acquired trait because it definitely can be inherited into the next generation. Maybe a lot of those traits, racial specific traits, maybe an epigenetic trait in nature, because the human genome project, when this human genome project was finished, now the next step is the scientists were very excited at the time, because once we have read all the alphabets in our whole entire genome, now is the time. And what's the difference in these alphabets among different races, like blacks and whites and Asians? Are there any some specific DNA sequence variation which represent some the characters, race specific characters? Scientists, actually majority of the scientists believe that there should be some DNA sequence changes, because after all, DNA sequence change in the form of mutation is what makes this all different traits. But then, to the surprise of all these scientists who have actually anticipated such, there are not really any significant and meaningful sequence differences which is specifically related with these race differences. So in other words, without having any particular outstanding sequence variation, all these different human races have maintained such race specific traits, like blacks have this darker skin and white is a white skin. And not only that, we all know that some race specific different, all different traits, but then none of them can they find any like meaningful particular related correlationship, correlated DNA sequence change. So then how else can you explain this? Maybe this epigenetics is the only alternative that you can use in the explanation. So the point is, not all but some epigenetic properties can also be inherited sometimes. So with that point in mind, maybe that the inheritance of the acquired traits, I introduced this in the first chapter, which was claimed by that John Baptist Lamarck and earlier than the Darwin's theory. Maybe not really such a incorrect idea after all. Is so everything's getting around circle. So that's the brief account for the difference between, in terms of this chromatin structure, eukaryote, euchromatin and heterochromatin. And here the important point is why euchromatin has such a high gene expression activity versus heterochromatin has a poor gene expression ability. It's all due to the compactness of the area. If the area is highly compact, highly condensed, once again, then all those require the factors, including the enzyme involved in the RNA making, and then later we are going to see actually there are many additional protein factors that actually are required for the enzyme. By the way, the name for this RNA making enzyme is RNA polymerase. That the two RNA polymerase to work, there are several different protein factors are also required, and they have difficulty if those area having that particular genes are organized in heterochromatin, and they cannot really get through this because they are all covered with the nucleosome, basically the histone proteins. So you have to You have to get rid of them before they can start working on this. Okay, so with that, I can finish this week's lecture, and actual the control of gene expression will be continued on next week.